Professor Saverio Gentile. Targeting potassium channels reveals weaknesses and strengths of cancer from biochemical signaling to precision medicine. Over to you, Savi.
Yes, hi. Thanks a lot for the invitation. I'm always surprised how people are really good in butchering my name. Thank you very much for doing this as well. I am very, very pleased to be in this meeting, and although it's virtual, I still consider this one of the best meetings for this type of interaction, and I've learned a lot already. So I hope I can be up to the task. What I'm going to tell you today is what we have found by manipulating ion channels, specifically potassium channels, in cancer, and we're focusing today on the breast cancer specifically. So we're going to look a little bit of how pharmacological targeting of potassium channel can affect several hallmarks of cancer, but also how can we use now potassium channel for therapeutic purposes. And so my overall idea starts from this very simple concept. Ion channels have been found in a variety of cells, and in some cells like neurons or muscle, they control very, very fast events like action potentials or muscle contraction. But they also found being important for controlling cell signaling and cellular events that takes longer than just the millisecond time scale in which we normally used to look in neurons. And I'm talking specifically about voltage-gated ion channels. And more recently, this voltage-gated ion channels appear to play a major role also in proliferation, which is, of course, takes hours of days in cells. So my interest basically in cancer is very arrogant in a way. I want to try to put all this knowledge together and try to figure out if it makes any sense in cancer biology. But also, as I mentioned before, how can we actually use this ion channel for therapeutic purposes? So ion channels have been found dysregulated in a variety of cancers, and cancer of different histogenesis, of cancer of the same histogenesis, they can be characterized by different, they have different molecular characterization and so on. So from neuroblastoma all the way down to liquid tumors, and some ion channels are particularly important for women, some other are devastating diseases for men and women. So there are many questions we can ask: why these ion channels have been dysregulated, why cells need these channels, what do they do, and again, how can we use these proteins? Can we actually target them, and how can we target these proteins? So what today I want to talk with you, it's examples of how targeting ion channels with drugs, with molecules, reveal several aspects of cancer biology. In some extent, we can see how, for example, stimulating specific potassium channel arrest tumor growth and metastasis, and in particular, I'm going to focus on what could be grouped as a gynecological cancer, specifically breast cancer, and more typical of women cancer, the ovarian cancer. So next, let's start from the breast cancer, which at this point I believe is a nice continuum of what we heard previously. So the breast cancers, we should use the plural for this type of cancer. It's generated or stems out from the breast, and can be characterized from the histological properties, but also molecular characterizations. And we can call them estrogen and progesterone receptor positive because they express estrogen and progesterone. Can also call it HER2, which is another surface membrane protein in cells that do not have estrogen or progesterone. And then there's a third category, which is kind of weird category, because we classify this cancer by not having either the estrogen receptor or progesterone or HER2, and so it's like asking somebody's names, what's your name? And the answer is, I am not Saverio, I am not Amanda, I am not. So that's a little bit interesting, at least from the biological point of view. But what we found more interesting is that the KV11.1 channel, which is encoded by the KCNH2 gene, or more famously known as the hERG channel, is expressed in all 3 types of breast cancer. And that has several important points. One interesting thing we found is that if you take the patients that express this channel and you separate the cohorts of patients, the one that has high expression of the channel, as you can see in the panel on the overall survival, these are the patients in the red line. You separate this patient from patients that have low expression of the channel. Then you see that patients with high expression of the channel, they have a better overall survival, which practically speaking means they live longer. And so, which implies that the more channel, the better it is. And so that could probably be meaningful also from the therapeutic point of view, because now we can think of stimulating the channel activity, and the stimulating the channel activity may actually have an effect on cancer. So what do you see in the left panel here? These are nude mice in which we have introduced subcutaneously triple negative breast cancer. Again, these are very, very aggressive cancer and do express the KV11.1. And you see these 2 big balls at the bottom under the control is actually indicating the size of the tumor after 18 days. But in mice that have been treated with the fairly selective KV11.1 channel activator, which is called NS1643, see the tumor in this mice is much, much smaller. And they gave us the idea that the stimulation of this potassium channel may actually lead to an arrest of the tumor growth. And the panel you see in the middle is more or less the statistics. And we repeated this experiment a variety of times. And also, we understood that the stimulation of the channel also affects metastasis. And actually, to anticipate Mustafa's question, I would say that the inhibition of metastasis actually happens because cells are severely affected. The cell motility is severely affected, and we are studying now the signaling that relates to that. But the overall idea is that stimulation of a potassium channel such as the KV11.1 is actually has a very strong effect on cancer growth. And as we have heard previously from Sarah Lynn, and we all know already that this channel is expressed in the heart. This was a kind of interesting idea, stimulation of the channel, because we all know that hERG channel is very sensitive to a variety of drugs, but the most important effect of this drug are basically they block the channel. When they block the channel, this event can associate with slowdown of the repolarization phase of the action potential and eventually increase of torsade de poids and ventricular fibrillation. So in our case, we're doing the exact opposite. So we're stimulating the channel, and from the tests that we did with electrocardiogram, imaging, and so on, at the concentration that we use for in mice, in which we do see shrinkage of the tumor, we don't see significant side effects either on the heart or kidney or other major organs. So the stimulation of this potassium channel doesn't seem to be as bad as blocking this potassium channel. So we used several models now. The one that you see on the left, these are patient-derived tumoroids. These are piece of tumors, basically from specific patients. These are triple negative patients. And with which we can actually do pharmacology, and we tested now a variety of channel activators. As you can see here, we have the NS1643, which is the one I showed you before, but also other like PD118057, and we have a long collection now that we're testing. And in all circumstances, what we see is that the stimulation of the channel, the baiting basically this tumoroids with channel activators leads to a shrinkage of the tumor. And interesting enough, if you see the one in the panel in the middle, the shrinkage of the tumor reaches a steady state. So after, in this case, under 20 hours, we don't see any further change of this shrinkage. So that implies that the tumor shrinks and then stops doing. And now we want to understand what actually is affected in these cells. So from the cellular point of view, when we look at the cell cycle analysis, we do see that the cells, the cancer cells, and that honestly happens in triple negative as well as estrogen receptor positive, and also other tumors like colon cancer we recently found. They also express the channel. They all behave in the same way. They arrest the cell cycle in the G0, G1 phase. And so we started looking now. So if we have an arrest in G0, G1 phase, do we actually have a cytostatic event, or that associated also with cytotoxic event, meaning are the cell actually dying also or not? And when it turns out that when we do, for example, on Nexin-5, but again, this experiment has been repeated with also other markers for cellular death, we don't really see an increase in cell death. So it turns out that the cells are stuck in G0, G1, they do not die. And if you do an experiment like this, you see on the left, you treat the cells for, let's say, 24, 48, and 72 hours, you see in the upper left panel, and you see the proliferation rate is strongly decreased compared to the control. But if, for example, in 96 hours, you remove the medium and you wash the cells every time, the cells don't go back in the growing phase. Now, the arrest of the cell cycle, actually, if I can share my video, my monitor here, let me try if I can do that. So this actually is a movie. How do I do this? It's a movie. So these are triple negative breast cancer cells that were infected with a virus that encodes for constructs that makes the cells green when they are in G2M and red when they are in G0, G1. So you treat the cells, and I hope you can see the movie, but if you treat the cells with NS, what you see is that all the cells basically become red. So they are stopped in G0, G1. So which confirms what we were saying previously. I can't see my screen anymore. Okay, here we go. What confirms what we were saying previously, that the cells basically are stuck in G0, G1, and they are also have an upregulation of a variety of tumor suppressors that you can see on the Western blot on the left side, like P21, but also markers for what is called a senescent phenotype, like, for example, P16, or an upregulation of beta-galactosidase, and so on. So in other words, we do, by stimulating the potassium channel Kv11.1, we have an activation of a senescent phenotype, which by definition is considered a phenotype in which cells actually have lost the ability to proliferate. And the more brave scientists that really believe in senescence, they think this is a permanent effect. So cells will never be able to go back into growing phase. But again, these are cancer cells. We need to be a little bit more cautious with that. So, of course, there are a variety of signaling that are activated with the senescent phenotype, one of which is actually activation of autophagy. But we started questioning more and more in terms of time. So where is the first event that can be particularly important for the senescent phenotype? Because senescence happened about 48, 72 hours, but activation of potassium channels should actually trigger events that are much, much faster than what we actually have recorded in that particular time point. And so, as expected, the stimulation of the potassium channel drives a hyperpolarization of the membrane. It's actually dose dependent. But this hyperpolarization, so what we're actually doing here, we're removing a bunch of ions, potassium ions from the cell, but the abundance of potassium chloride inside the cell is so high that even if you remove 20, 30 millimolar of potassium, that most probably the cells will still have so much potassium chloride, and honestly, it doesn't really make a big change. But if we now associate this hyperpolarization, for example, with another event, which is a calcium entry, and again, I want to try to share the screen again and try to figure out if I can show you this other video. It says share. Okay, so what we have done in here, we have measured intracellular calcium concentration with a classical Fourier 2 before and after application of NS1643. And so what we do, basically, what you can see from the video is when NS is actually applied to the bath, and when the cells turn red, means that the calcium concentration, intracellular calcium concentration, has changed. And so what we later have figured out is that this calcium change occurs because we have an increase of calcium entry. And the idea is relatively simple. It's also been observed in immune system cells. When you stimulate the potassium channel in non-classical excitable cells, you drive calcium entry since the cells gets more hyperpolarized. And now that the calcium change in the cell, a lot of things can happen. And one thing that we noticed right away is that the mitochondria were actually severely affected. As you can see, using MitoTracker and also TMRM, these are two different ways for measuring mitochondria morphology and also, in some extent, function. So it only takes 5 minutes for the application of an NS1643, and to maximize basically this experiment, the effect, we use 50 micromolar. But if you reduce the concentration in a longer time, you see more or less the same effect. And what you can see from the MitoTracker is the appearance of these green blobs, which are indicating that the networking of the mitochondria is disrupted, and also this is confirmed by the TMRM. And also, if you can see, the mitochondria gets depolarized. And so the morphology of the mitochondria is severely affected by the stimulation of the potassium channel. And then if you repeat this experiment in the presence, for example, of EDTA or EGTA, you don't see the effect of an NS1643. So it is what appears to be an NS1643 driven calcium overload of the mitochondria. And if you look now at the function of the mitochondria with a seahorse analysis, and we used two different types of cell lines. These are MCF7, these are ER positive. They have the estrogen receptor cells, but the MDA231, they don't have these receptors. Instead, they're called triple negative. In both circumstances, you see that at different concentration of the NS1643, you see a progressive dysfunction of the mitochondria. The mitochondria doesn't produce enough ATP, and that's why we think is autophagy is actually now activated just to try to compensate the lack of ATP. But again, it's a topic for another discussion. The point here is that when mitochondria are failing, there is a very often direct association with an increase of superoxides. That's because the respiratory chain is out of control, and that's exactly what we measured. We do see that 2 hours after application of 1643, we have an increase of superoxide formation, and then 4 hours after, we have a further increase of superoxide formation. But then, if you look now at 24 hours after, when the cells start becoming senescent, you see that the concentration of the superoxide is very, very low. It's actually lower than the control, almost half of the control. And so we couldn't figure out what was going on, because you stimulate the channel, you disrupt the mitochondria, you create this ROS, and that potentially should actually kill the cells. But at the same time, we do see that the ROS formation have been reduced over time. So to understand a little bit better what was going on, we took advantage of our RNA sequencing that we had performed at the time. And what we figured out is that the most upregulated set of genes are actually the genes that are related to the oxidation reduction process, as you can see from the histogram, the bar graph on your left. And when you see on the right, it's just the fold change in logarithmic scale of the changes of these genes. Now, the vast majority of these genes, and the list is much, much longer, we go into hundreds of genes, are actually genes that are considered antioxidant genes. So we stimulate the potassium channel, the mitochondria is compromised, there's a ROS production that goes up in time, but then the cell senses this ROS production and try to compensate. They compensate by producing antioxidant genes. And that's a kind of exciting, but the question is, how do we even stop this process? Because now we are activating hundreds of genes. It turns out that the vast majority of these genes are actually controlled by one transcription factor. This transcription factor is called NRF2. And you see, interesting in this, you see in panel B in the Western blot, NRF2 is upregulated 2 hours, only 2 hours after that we stimulate the N-acetyl-43. But the downstream effectors are not upregulated. It takes about 24 hours for the upregulation of the downstream effector of NRF2. And that makes sense because we do see the downregulation of ROS 24 hours after the application of the channel activator. And indeed, if we now remove NRF2 from the cells, as you can see from this FACS analysis down at the bottom, we do see that cells without NRF2, they present a much higher ROS production. That makes sense because the cells are not compensating the normal ROS production. But now, if you apply an N-acetyl-43 for 24 hours, the ROS production is not stopped. You still have a lot of ROS ongoing. And indeed now, this doesn't show very well, but what actually you should, maybe the Western blot is more explicative, what you see is that 24 hours after now, the cells actually activate apoptosis. So while before this event was not happening because you did not have enough ROS, now you have enough ROS and the cells basically are dying. So that was kind of interesting. We have this series of mechanisms that cells actually activate in order to compensate whatever is the insult. So the cells get are going around without mitochondria, with not enough ATP, and yet instead of dying, they actually activate senescence. And in terms of mitochondrial damage, instead of dying, they're activating an antioxidant response to detoxify. I think this is quite interesting because not only we know that we can shrink the tumor by stimulating this particular potassium channel, but at the same time, not necessarily in a medical setup, in a clinical setup, we can also understand what are the mechanisms that the cells actually use to counteract this insult. And if we know enough, then we can interfere with this mechanism and eventually thinking of a dual approach with a combinatorial approach. And that's exactly what we're trying to do. But now, an interesting thing is that how can we use this from the therapeutic point of view? And all the drugs I told you before, these are drugs that have numbers. So they're not FDA approved, N-acetyl-43, the PD-11, and so on. So then I had a question, so can I actually find a cancer that has a dysregulated potassium channel, more appropriately, a downregulated potassium channel, that now can be targeted by an FDA-approved drug? And there are several cancers. The one I got fascinated more than the other at the time was the ovarian cancer. This is actually quite a bad cancer. The vast majority of the patients develop resistance to the current therapeutic approach, and the overall survival is really, really poor. But also in this case, as you can see from the bottom panels on to the right, the more channel, the better is the overall survival. The more channel, the longer this patient live. And the KCNJ11, or actually 6.2 channel, it has been found downregulated. And again, the more of this channel, the better it is. So the idea is, let's stimulate this channel. But how do we stimulate this channel, and what is the FDA-approved drug? So the FDA-approved drug is called Lonitin, which initially was patented as an antihypertensive drug. But then people that were studying this drug also realized that the mice that were used for testing, they had a very large fur. And so they understood that stimulation of the channel arrests hair loss. And so they patented under the name of Rogaine. The channel is relatively complicated. There is the tetramer that allows ions to go through. But in order to function, this tetramer needs the SUR subunit, which happens to be the target for minoxidil. So the patients are actually express this channel. It can be up or down regulated. But the experiment that we've done, we took a cell line, the OVCAR8, that do express the channel, and you can see in the upper panel, this cell lines are actually generating a very, very large tumor, and that's a typical of ovarian cancer type. It's highly invasive. It invades the entire peritoneum. And each one of these blobs that you see in here is a distinct tumor. So it's extremely invasive cancer. But in mice have been treated with 10 milligrams per kilo of minoxidil, present no tumor or very, very small traces of tumor, which indicates that the minoxidil actually was able to stop the tumor growth. Later on, we realized this is now a cytotoxic effect as the cancer cell actually die. And so the overall idea of this talk is that for treatment of cancer, we have a variety of options. Of course, we know there's surgery, targeted therapy. You can target estrogen receptor positive or negative. There is radiation, immunotherapy, chemotherapy. But each one of this present their own limits. For example, targeted therapy, you need to have a target. Otherwise, with a triple negative, you can't have a target unless it's the KV11.1 channel. Some other are invasive, some other are toxic. So what we are trying to propose here is really to target, to use potassium channels, but honestly can also be extended to other channels for pharmacological targeting, and offers a variety of advantages. One is the arrest tumor growth, or being also cytostatic or cytotoxic. And we have just recently got funds for activating a clinical trial on ovarian cancer patients that are resistant to carboplatin and cisplatin. And we're going to use minoxidil to see what's the effect on this tumor. So the overall idea, again, I want to reinforce this, using this particular approach, we can have an effect on the overall growth of the tumor. I didn't have the time to show you about the metastasis, the effect on metastasis. The cell signaling related to motility is strongly affected. But the point is that from the biological point of view, from the scientist point of view, we can actually study also mechanism that cancer cells use to counteract whatever is the insult. And in terms of antioxidant, if you think we hear so much about using antioxidant to be so beneficial, but now if you think about cancer cells using antioxidant response to survive, we should probably rethinking of using that much or that many type of antioxidant. And these are the people that are involved in this project. They helped me at different time of the study, and including Sofian, with which we're having a wonderful collaboration with Daniel Souther is patching for us cancer cells that really been done, and we're moving forward with this project really, really well. So I want to thank everybody in the audience, the people that helped me, and the organizer, and again, Sofian once more.
Thank you, Savi. Fantastic work. A really lovely story. Shame about my pronunciation of your name. I apologize for that. But keeping that theme of butchering Italian names, I'm going to go to our new application scientist who just joined us yesterday with a question. Beatrice Badoni, or in my English version, Beatrice Badwan. Have you looked also at extracellular acidification, ECAR, with the seahorse?
We are doing this right now, not only with the seahorse. With the aim is to exploit whatever would be the changes. I can't tell you the data because I don't have it yet, but I hope I can give you this information later on.
Okay, and we'll move on because you've got a lot of questions, Savi. We'll try and get through as many as we can. I mean, we're at the end of the talk, or end of the session, so if anything, you know, hopefully we can go on, and if people stick around, we'll keep asking questions as long as Savi's welcome to stick around. So Nazreen asks, is it known if the expression of KV11.1 changes as cancer progresses?
That's a great question. We don't know yet. At least I don't know yet. I know there's other people also working on the KV11.1. We're also focusing, though, on other channels. And although the KV11.1 has been found, the expression level from the mRNA level has been found changing during the cell cycle, in different cell cycle phase. I think Arcangeli showed this in a paper. We could not see this translating in change of protein. But if you look at other channels, we start seeing these changes. And the reason in our case is because this other channel we're focusing is controlled by estrogen. So there are possibilities for other channels to be changing during different phases of the cell cycle, or different phases of the cancer progression. Again, these are questions that need to be explored a little bit better, and it will take a little bit of time.
Okay, and Jonathan Sobolov asks, what links KV11.1 activity and mitochondrial function?
Not that they're, sorry, can you repeat the question?
Yeah, sorry, it's what links KV11.1 activity and mitochondrial function?
Okay, so first of all, hi John, how you doing? Second is the hyperpolarization as a consequence of potassium channel stimulation drives calcium entry. So this calcium overloads the mitochondria. this is probably the most important effect. However, there is some literature describing Kv11.1 on the mitochondria. However, I'm not sure that this particular aspect is relevant for what we do, and the reason is because the drug that we're using acts on the extracellular domain of the Kv11.1, but things need to be explored better.
Okay, and Sarah Lynn asks or states, very interesting talk. Do other KV channels other than KV11.1 play a similar role in these cancer cells? Is there hope for us in the KV7 field?
Well, the simple question is yes. We have identified some of the KCNQ family, and that is the reason why I was asking her if she was interested in testing her drugs on cancer. And I think it's going to be important because it's there are 80 genes that control potassium channels, but few of them are significantly dysregulated in cancer. Among the KCNQ family, there are some that are really interesting, and then there are some other TRIP channels as well, but these are totally different family.
Okay, so there's hope for you as well, Sarah. Get on to Savvy and you might be having a collaboration before you know it. Emma Veal asks, is KV11.1 expressed on mitochondria in these cells, or are the responses subsequent to plasma membrane hyperpolarization? First part of the question. Do you want me to give you the second part, or do you want to take that first and then we'll move on to second?
Yeah, no, go on with the second, please.
So the second part is, if the latter, is the effect selective for KB11.1, or can other K channels mediate this if expressed?
Okay, so I think I mentioned earlier that there is literature describing the KB11.1 on the mitochondria. However, and it might play an important role. Honestly, I need to see more data to be completely convinced that this is true. It was done with using antibodies and a variety of other things. I don't know if there is any electrophysiology on mitochondria, catching some hERG channel. The drug that we use, the NS1643, but also the other drugs that are chemically distinct, and we have now 6 or 7 distinct drugs, are acting all extracellularly. So I don't think there is any chance that the drug permeates the membrane and hits on the mitochondria. However, having the channel, assuming that it's true that the channel is on the mitochondria, there might be a chance that the channel responds to the change of ionic gradients in a way or in another. The same literature also describes the KV11.1 activity on the mitochondria in the cardiac myocyte being protective for the cardiac myocyte stress that these cells might undergo. So it may actually be even an advantage of having KV11.1 on the mitochondria because it protects the heart from stress like ROS-related stress and some other.
Okay, we've got a couple more. So if you're willing to stick around, we'll carry on. Hi asks, what is the effect of the opposite action by inhibiting K channels in cancer cells? Does it make the cancer worse?
Not necessarily. This is an interesting thing, and we were just coming out with a review trying to explain why these proteins are important. So let me go into the details. If you block the hERG channel, you can actually kill the cancer cell. And blocking the hERG channel, by the way, is not similar to stimulating the channel. Blocking the hERG channel also includes removing the channel from the membrane, and you removing a protein that potentially can bind other proteins and so on. The reason why this particular approach is not okay, while this is actually confirms that the channel is important in the cancer biology, I like to point the attention to the therapeutic use of this approach, and you might actually risk to increase events that are already dramatic for normal people, but when you are in a cancer setup patient, then you're talking with a patient that's already undergoing a bunch of other stresses and so on. You don't want to touch the heart very much. Did I answer the question?
Yeah, I think so. I mean, yeah, that was a pretty thorough answer, I think. So Scott asks, any known interaction of KV channels and mitochondrial CB receptors?
mitochondrial CB receptor.
Yep.
I would need to know a little bit better what is the mitochondrial CB. I'm ignorant on this, and I apologize.
Me too, to be perfectly honest. I'm kind of thinking cannabinoid, but
Yeah, I would like to. Yeah,
endocannabinoid. Scott does confirm, yeah.
I honestly don't know the answer. I don't know the literature, and I haven't explored. But now you made me curious. I'm gonna look into it.
Well, again, that's hopefully what these meetings do, right? Is they stimulate interest and new ideas. And I was watching the chat earlier, and it was really nice seeing some of the interactions. Anyway, moving on. Jean-Francois asks, thanks, Saverio. Terrific talk. Going downstream, do you think the mitochondrial calcium uniporter is also a valid target?
Yeah, I think so. This is not my direct field, but one of my collaborators back in Italy, Luigi Leanza, is actually working from the Zabo lab initially. Now he has his own lab. I think he's looking into that, and it's quite promising as well. So I would suggest to look at his literature. He can provide much better background and details than what I can do.
Okay, and I think this might be the last one. So if anyone's got a final question, you better get it in quick. John Soboloff comes back again. Are these store-operated calcium channels whose activity is increased by hyperpolarization? And the second part, can you protect against calcium overload with calcium channel blockers?
Okay, the calcium channel blockers, as John knows, are not super specific. I can tell him right away that if you remove calcium from outside, even by using media with no calcium or just adding EDTA and GTA, you don't have the calcium overload. About the store-operated channel is something that we would like to look into it. The initial experiment that we've done by using the conventional blockers don't seem to suggest that the store-operated channel might be directly involved. But again, this is still too superficial to be conclusive, and it needs to be explored in more detail.
Okay, I think that wraps it up. So great talks by all the speakers today. Thanks very much. We all really appreciate your time and your efforts to put these talks together.